Thank you, Rita, for the very nice introduction, and thanks to all the organizers for inviting me back to Bologna, although this year is only a virtual presence. But I must confess that I'm very fond of participating in the Bologna Winter School for a purely personal and historic reason, which is well explained in this picture here. This is actually how it all started. This was the first, at that time it was Bologna Summer School, but I think it had a very fitting acronym, which is BOSS, and it was in 1999. And this was actually the first summer or winter school I attended during my PhD at that time, when I was still in Germany. You can see that we were all much younger at that time. That's myself, that's Rita, that's Gigi Martelli, and there are some of the some other people you can recognize in this picture. And well, it had a deep impact on myself, as it basically got me into the whole field of protein structure prediction, which is something we have been doing ever since. We have been participating in CASP for a number of editions, and then more recently also other challenges. And the latest edition, which I'll be touching on today, is that we designed our own challenge for assessment. But so yes, I have very fond memories. I've been back to Bologna a number of times, and it's always always feels like coming back home. So today, what I want to talk about is a topic which was initially considered to be more controversial. Now it's becoming more mainstream. But the key point I want to make about why to look into this kind of phenomena is well captured on this slide here. So over the last several years, we have been dealing with a veritable data deluge. So this is basically one of the side effects of having next generation sequencing and all those sequencing machines which are churning out ever more sequence data. And especially for all the various organisms for which the sequencing has been produced, this has led to the accumulation of an exponential amount of nucleotide sequences, which can get then translated into protein sequences. And in fact, what you see down here is the growth of the UniProt KB database, which is the one repository where all the known protein sequences are hosted. And you see that this is essentially exponential. There was this bump here in the middle, but this is because it was already growing too fast. And then they, at UniProt, they decided that their model was no longer scalable, so they had to reduce the redundancy in the database. So they saved a bit, but then we're back to exponential growth. So as of this morning, this is the content of the UniProt KB database. So we have actually two components, which is SwissProt, slightly more than 500,000 protein sequences, and these are the manually annotated and reviewed entries. Sorry. So these are the sequences for which there is actually knowledge in the form of an expert who has looked at the data and has validated everything which is listed on the UniProt webpage. On the other hand, you see down here that TREMBL, which is just TREMBL is the acronym which stands for Translation of EMBL, which was at that time the nucleotide sequence archive. So TREMBL contains over 209 million sequences. And these are only automatically annotated and are not reviewed. In other words, any knowledge for those over 99.9 or so % of the sequence, protein sequences we know of are only deduced from algorithms. And therefore, this means essentially two things. First, that you always have to treat the information which comes from TREMBL component with a bit of extra care because it may contain errors. But the second point is also that any increase in the performance of the algorithms which are used to annotate protein sequences in the Tremble component of UniProt, even if it's just 1 or 2%, will have a tremendous knock-on effect, because you're talking literally about millions of sequences where that added annotation can be produced. So this gives you the kind of large scope we're dealing with, and why it's important to work with computational methods to try to annotate these protein sequences. And now let me tell you a few things about how the databases, especially UniProt, is built. I've already mentioned that there are 2 components to this, and the really key point here is what happens in Tremble, because this is just the automatic annotation. Sorry. So all the sequences which are in the nucleotide databases down here, be this ENA, GenBank, or DDBJ, these are entered into UniPark, which is a sequence archive. And this sequence archive is then the basis for UniProt. And you may ask yourself, why do we need a sequence archive? Shouldn't the sequences be unique? Well, over time, as strange as it may seem, some sequences have been revised. Maybe this had to do with sequencing errors. Maybe it came out that one particular isoform is the dominant one, or other changes. So there is the separation between the archive and what is available in UniProt KB. This also ensures greater reproducibility. But the key point here is that the knowledge in UniProt, which is the part here in the middle, stems essentially from a few sources of information. One of them is manual curation. This would be for the SwissProt component, obviously. So it's not available for the vast majority of sequences, although the most relevant human sequences fall in the category which, of course, has manual curation. But then what we have in terms of automated annotation is essentially shown on the top part here, where you have various databases which contribute the automatic annotation. So the ones listed here, so Gene Ontology would be for functional information, Interpro is for protein families and domains, Intact is for protein-protein interactions, and a couple of others. But really the mainstay of how the annotation pipeline inside the EBI, which hosts UniProt, especially Tremble, works, is that Interpro is the key to the annotation for all new sequences. And Interpro tries to leverage the concept of protein families and domains. And so most of the information for all the sequences which don't have the manual curated component comes really from Interpro. Now, Interpro is a database which essentially is a federation of various databases which have grown over the years and which all annotate in a very similar way. So the key concept here is the protein domain, and protein domains are seen as distinct functional and or structural units in a protein. So a good example here would be this small SH3 domain. This is the structure. And if you draw it in a simplified way, the protein sequence, then in this case, we would have 3 consecutive SH3 domains plus an SH2 domain towards the C-Terminus. So the protein domain concept comes in very handy because it gives you the equivalent in protein sequences of what would be a Lego brick. So it's a module which can be reused in different contexts. It's an evolutionary concept as well, because these are sequence families which can be transmitted among different organisms. They can also exist as paralogs, so as slightly specialized versions in the same genome. For instance, SH3 domains in the human genome, there are scores of proteins which contain this domain, and they all perform to a certain degree different functions. But this module is repeated and can be identified more easily. And then, of course, there's a classification element. So you can try to break down the protein domains into groups of superfamilies, families, and subfamilies. So, of course, then this comes in very handy. It is also easy to detect these domains because you can recognize them from the sequence alone. You only need to know that they correspond to something which has a functional or structural meaning, but they can be identified with a simple sequence alignment. So Interpro then, in reality, distinguishes between domains, which are these functional modules. They also have families, and families are the ones where it's not clear whether it is a true domain in the structural or functional sense, but it certainly is a group of sequences which share this region of high conservation, which is homologous, but it's in a way a weaker statement compared to domain. And then there's a special subtype I won't go into, which are repeats. This is another of our research interests, but it's outside of the scope for today. And then they have annotations for sites, which can be things like active site, binding site, of post-translation modification site residues, which are annotated. As I was saying, Interpro is a federation of different databases. We have our own contribution to the Interpro consortium, of which we're part for a couple of years now, because the Mobidb database, you'll hear more about that later, forms part of Interpro. And more recently, we've also become part of the PFAM consortium, contributing to the annotation. So Interpro gives you different views of the same sequence in a way. And the component databases, well, they are curated to different degrees, in the sense that some of them are quite large. So PFAM is one of the largest contributors, alongside Panther and a couple of others. Some databases haven't been really funded so much for the last couple of years, so they are there because they were there also previously, but they are smaller. And basically, all of them, they share this concept about annotating protein domains and recognizing them, etc. So to just tell you briefly how it works in PFAM, since this is pretty much the mainstay for the annotation of novelly sequenced genomes, for instance. PFAM specifically is a collection of multiple sequence alignments based on hidden Markov models. So what you can imagine here is that you have curators who believe that a given sequence fragment is relevant. Then they collect information from the literature about what may be the size of that domain. They add additional sequences to the seed alignment. And then this is made into a hidden Markov model, searched against all the other sequences, and some of potential overlap issues are resolved, and the size is optimized, and then this becomes a PFAM model representing a given domain or family. And this is then also annotated in terms of a description about the function associated to this domain, or if the structure is known. So this is really what is going on in order to build these domains. Domains are a very powerful concept. With domains alone, you can annotate at least half of the residues in the human genome. And there were especially some early on some very good success stories where you could identify these modules and see them to be used evolutionary in different sequences, and therefore add a lot of functional information. However, with with many concepts in biology which are successful, they are successful up to a certain point. And then you start to wonder whether there's something else or whether there are limits to this paradigm. Specifically, there is this paper which came out in 2013, which has a rather technical title. And in fact, it's also in a rather technical journal, which is Database. The title is The Challenge of Increasing PFAM Coverage of the Human Proteome. And this is taken from the angle of PFAM trying to improve its coverage of the sequences, but it contains some very interesting remarks. And one of them is that there may be something else out there besides protein domains. So one of the most important figures in the paper is shown here. This was figure 1 from the paper, where the authors looked at 3 model organisms and the degree to which they were annotated with PFAM at that time. So for human Baker's yeast and E. coli, in blue, you have the fraction of sequences which have at least one PFAM annotation. And unsurprisingly, for human, 90% of the sequences have at least one PFAM annotation. So it means that more or less the domain, the paradigm of using protein domains works well. However, the first point to notice is that about 10% of the sequences do not match any PFAM domain known at that time. Now, this may be because the PFAM curators didn't have enough time or enough resources to curate novel families, or it may point to something different. And the fraction here goes down for Baker's yeast and is again over 90% for E. coli. In Baker's yeast, it's already about 25% of sequences which miss this annotation. So again, these may be domains which are not properly represented in PFAM. However, if you look, and these are the red bars, into the fraction of residues which are covered by the PFAM annotations, then you see that for human, at that time, it was less than 50% of the human proteome having a PFAM annotation. So, in other words, slightly more than 1 out of 2 residues in the human proteome, they are there, they are conserved usually, they perform some kind of function, most likely, but they don't fit into the PFAM domains. So this is a stronger indication that there is probably something else out there. This fraction is roughly similar for Baker's yeast, and it's to a certain degree higher for E. coli, but then E. coli is a simpler organism, and therefore it's easier to detect the enzyme and to measure enzymatic functions for the protein, so this may contribute to this higher fraction being correctly recognized. Now, the second element from the same paper is this table here, where the author looked at possible interpretations of what was missing out there. So at that time, PFAM still had a division between PFAM A and PFAM B. PFAM A was the manually curated, which is still maintained. PFAM B was discontinued over time. So never mind this column here. The important thing is between the first column and the last column in particular. So the last column are the things which are neither PFAM A nor PFAM B, and they are not signal peptides. Signal peptides because they can be recognized, and they are of course not a domain, but they perform a specific function. And these regions should be at least 50 amino acids long. So a couple of possible explanations at that time. One, that they could be coil regions, and yes, there is a bit of those across the board, but it doesn't contribute too much. Really, what was the main culprit they identified was listed here as disorder. So there is a concept of intrinsically disordered protein regions, something which is still called in certain areas more flexibility or conformational variability, but more about that later. And this seems to be alone responsible for some 40% of the missing residues which are not properly annotated. Then there are low complexity regions. So these would be things where you have the same amino acid type multiple times. Think about polyglutamine, for instance. And there's relatively little in terms of transmembrane regions. So there is a clear indication that the phenomenon known as disorder could be the major culprit for the missing annotation in PFAM. And in fact, at around the same time, well, 1 or 2 years earlier, there was this editorial coming out of Nature, which has been summarizing a number of developments in the field of protein science over the previous decade, which says breaking the protein rules. You remember that the traditional paradigm for protein structure and function is that the protein sequence entails one native state, and that native state conformation of the protein is the one responsible for the function. This is clearly the case of enzymes, for instance, and this has been the dominant paradigm for a few decades. However, by the time this editorial came out in Nature, there were already a lot of indications that if dogma indicates that proteins need a structure to function, then why do we have so many concrete biological examples where the function is carried out in a state of disorder? So something was missing there. And the first indications that there was something missing already date back to the late 1980s, where Paul Siegler, in this very short piece, was arguing about acid blobs and negative noodles in transcriptional activation. So clearly there were parts of the protein sequences which were not properly folded, and they were the ones interacting with DNA. So something special was going on there. Of course, it was not called disorder at that time. Then later, one of the milestones in the field was this review published by Peter Wright and Jane Dyson in JMB in 1999. And this was a milestone precisely because Peter Wright and Jane Dyson were NMR spectroscopists. And so they had all the intentions to try to study proteins with, from a structural point of view, with NMR techniques and other structural biology techniques. And yet they found some proteins which they were not able to treat with a regular paradigm. And so this first review is just a collection of cases where they found out proteins which were probably unfolded and yet were carrying out a function. And therefore, the conclusion from their abstract was that the assumption that a folded three-dimensional structure is necessary for function needs to be re-examined. Of course, this is a collection of a handful of biological examples. And how do you generalize from there? Well, this is an area where bioinformatics has been a driving force. And to jump some years forward and to give you also part of the answer to what the purpose of disorder is, here are some, a table and a figure which come out of two separate computational studies. This table here comes from a paper by David Jones. Again, a JMB paper where he developed the Disoprad predictor. And then he went on to see how much disorder was predicted in the different kingdoms of biological organisms. And it turns out that both archaea and bacteria have very little predicted disorder, on the order of a few percentage points, but that eukaryotes have a much larger fraction. And specifically among the eukaryotes, it's not shown here, the higher eukaryotes and the mammals in particular have an even higher fraction of proteins predicted or regions predicted to be intrinsically disordered and to be long. So something was going on there. Another possible explanation here comes from this paper from Burkhard-Rost's group, again published in JMB, where they looked at some functional classes in these pie chart, double pie chart diagrams. And essentially, this is the simplified view where they only look at three different functional classes. It turns out, so what they call Norse proteins is what can be roughly translated as intrinsically disordered. And what they call non-Norse proteins would be everything else. So the proteins which we expect to be behaving according to the traditional paradigm. And what you see here is that the broad category of communication is enriched for intrinsically disordered proteins. And in particular, the regulatory functions seem to be an area where it is advantageous to have intrinsic disorder. Again, this is all based on predictions. So it has to be taken with a grain of salt, but it gives you a rough feeling that disorder is a feature correlated with the organism's regulative complexity and the communication it requires. So something which was apparently used over evolutionary time, especially for the higher organisms. Now, but what does it mean, or what can we expect from regions which are either predicted or shown to be intrinsically disordered? Well, let's take an example you probably have already heard, which is the p53 tumor suppressor. p53 is also known as the guardian of the genome because it's a master regulator responsible for inducing apoptosis and preventing the cell from developing cancer. p53 is a short protein of less than 400 residues, and yet this is the interaction graph for p53. So you can clearly see that we have a very nice and large hairball of proteins which interact with p53. The number, you have to consider that the number of proteins which are known to interact with p53 is by now, I think, over 500 or so. And contrast that with less than 400 residues of protein sequence. What this tells you is that if all of those proteins were to interact with p53 simultaneously, they would be fighting over slightly less than one residue where they could be binding. So clearly there is something where there is regulation going on, but there must be also sites where multiple proteins can interact at different points in time. And in fact, here you see a diagram with a prediction. This is the Ponder score, the prediction for disorder, from the N-terminus to the C-terminus. A high Ponder score, so towards 1, means that it's predicted as disordered. A low Ponder score below 0.5 means that it's predicted structured. And here you see with these colored bars a number of pieces from p53 which were found in protein structures deposited in a protein data bank at that time. So crucially, the central part of p53 is known to be the DNA binding domain, and it's shown here in pink and bound to DNA. And then we have these other parts at the N-terminus and the C-terminus. And you see that here there are short fragments of p53 which are co-crystallized with other globular domains. And these are consecutive and they fold up in roughly similar shapes. So it tends to be an alpha helix. So this could suggest that maybe that stretch is just a longer alpha helix and you can have different proteins binding that, apparently. However, the C-terminus of p53 is where things really become much more complicated. Look at this short stretch here, which is about 10 or so amino acids, and it's shown with different colors. And you see here that depending on the binding partner it binds to, it adopts completely different structures. So in one case, here it's irregular. It looks a bit like a loop on the surface of this binding partner. Then in this case here, when it binds sirtuin, it becomes a short beta strand in order to fit into the binding pocket. And when instead it binds S100 beta beta, it forms this short alpha helix. In other words, this little tiny piece here of the p53 protein sequence is able to adapt to the binding partner, which just happens to be around at that time, and to bind it, adopting different conformations. So this is clearly a region which is carrying out its function with different structures, and it's probably there when it's alone. It probably lives in a conformational continuum, moving between this and probably also other conformations, according to some thermodynamic equilibrium, which can be shifted. So this is one of the examples of what you can expect with intrinsically disordered regions. To see a bit more in context, here you see a number of cases. This slide was originally from Toby Gibson at EMBL Heidelberg, where he was mainly concerned with the protein architecture modules in cell regulation. And you have about 3/4 of them which are globular in type. So these follow the traditional paradigm, and they are effectors of regulation. Sorry, today my voice is terrible. And on the other hand, you have about 25% of the sequence, which is thought to be natively disordered and can be broadly seen in at least 3 different types. So this can be either things like the p53 tetramerization domain, which folds up in these nicely ordered helices and beta strands and forms the tetramer because each of the single protein chains contributes one such element. Or you can have here cases of induced fit where we have a longer stretch which essentially wraps around the globular domain. This is the case of the SARA protein. Or you can have short linear motifs which binds to things like SH2 or SH3 domains, and this is just on the surface of the protein. All in all, these tend to be the passive components involved in building regulated and often highly dynamic complexes. So these are the things where really something goes on in terms of structure, and they have to adapt. This type of consideration has been carried forward, and there is this interesting paper from 2014 by Tompa et al., where they tried to estimate the number of different cases you can have in the human proteome, where it started out of the entire human proteome about 3 to 4 million residues. That's about 1/3 of the entire proteome is intrinsically disordered. And that, if you look at the various cases and instances you can have for protein-protein interactions, well, then at one end of the spectrum, you have the traditional protein complexes, which are large. They involve very long proteins, so that's few of them. The typical length in terms of amino acids for the total complex is over 1,000 residues. And there is thought to be between 600 and 1,000 of such complexes in the human proteome. Then, smaller than those, you can have the globular domains of about 100 residues, a typical one in length, and these may be something like 35,000 in the case of the human proteome. Shorter than that, you have the binding motifs, so things like SH2 or SH3 domains, where you have a peptide from one protein which binds to the globular domain here. The typical length of the peptide being bound is about 10 residues. And these are already thought to be on the order of 100,000 instances. And the smallest type of sites you can have are post-translational modification sites, things like a phosphorylation site. And in this case, it's the length of the interaction site is 1. And the estimated instances in the human proteome is about 1 million. So this may be an upper bound estimate, but even 100,000 transient, conditional, and regulatory interaction modules is quite a lot of things, and we are still only scratching at the tip of the iceberg. So there are a lot of these interactions out there, which are also hard to define from an experimental point of view, and therefore these are instances where the intrinsically disordered protein regions are relevant. Consider also that disordered regions cover about 70% of all experimentally validated post-translational modifications. Phosphorylation sites in particular are highly enriched in disordered regions, and they are quite rare inside globular domains. So this is just a sort of overview of what you can expect. But so far, I've only been telling you that there is something called disorder. Yeah, but what does it exactly mean? So intrinsically disordered proteins and regions can exist in a variety of different ways. So on one end, at one end of the spectrum, you can consider them to be a bit like a cooked spaghetti in water, which is just floating around and moving randomly, and there is no secondary structure left of any kind which you can detect. And there are instances where this is probably the case. Then there are things which are disordered, but they are compact. So you don't have much in terms of secondary structure, but they behave more or less like molten globules. So they move around. It's not a fixed structure, but yeah, they are compact. They are not extended, at least. Then you can have instances where the disorder is somewhat rigid, in the sense that you don't have the full dynamics which you had in the beginning here, but they are prevalently extended, and there's not much in terms of secondary structure. And finally, you can have a category where they are still considered disordered because they're moving around, but they do maintain secondary structure elements. So for instance, an alpha helix, or something which may become a beta sheet under certain circumstances. But especially shorter alpha helices can be present even in the disordered state. Of course, if you notice, this is just a schematic drawing, because a lot of data in this area is missing, and it's very difficult to capture really the structural ensemble. So we're going in a way by inference. We're trying to identify what is disorder by also stating what it is not. And so it's about trying to figure out really what is in there. And this is still one of the major challenges. So to give you an example of a protein which is intrinsically disordered, this is the protein Drosophila mastermind. And notice that it has a very strange amino acid composition. So you can tell by just looking at this protein sequence that there is something strange going on. And yet this is a functional Drosophila protein. So it does something in the Drosophila cell. Otherwise it wouldn't be there. It would have been eliminated by evolution. And yet look at it. There's these long polyglutamine regions here, a lot of Qs. You have other regions where it's polyasparagine, here a lot of Ns. Then you have regions here which are rich in glycines. You have other regions which are fairly normal in the sense that they have different types of amino acids. So it looks a bit like a mess. And what do you make out of that? How can you try to infer anything related to the function of this protein just from the sequence? That is one of the questions here. But even before we try to address that, let me say a few words about how you can predict disorder of something which approximates disorder. And in fact, one of the earliest attempts to identify something which is not quite right, so to say, was the algorithm SEG developed by Wooten in 1994. For the record, SEG is usually used as a pre-filter for blast searches against the database, because it identifies regions which are like these ones here in Drosophila mastermind, so these poly-Q regions. If you use a blast search on one of these regions, you would pick up a lot of other sequences which are not really related by evolution to yours. So SEG was an idea to filter out those more or less easily. So the low complexity regions here are identified according to this formula, which is just Shannon's entropy. So it measures the Shannon entropy along the protein sequence. And here is part of the original paper where they try to look at different data sets and to see whether there were any low complexity regions out there. So let's start with the PDB. If you use the SEG algorithm on all the sequences where you have PDB structures, you end up with this distribution here. So lower values means that you have fewer types of amino acids which are enriched in longer stretches. And if you shuffle the PDB sequences, you get this distribution here. So essentially it tells you that there's probably nothing strange going on with the proteins which have been crystallized and deposited in the PDB. Now, if you do the same analysis for SwissProt at that time, and this were the early 90s, so it was a much smaller database than today, you get this distribution here with a very long tail towards low complexity values. And interestingly enough, if you shuffle the SwissProt sequences, then you see that you have the same distribution as you had for PDB. In other words, it is not a feature where the amino acid composition per se is different in SwissProt. It is really that there is something which is conserved in the sequences and which does not behave in the same way as what you can find in crystal structures. Over here at this end of the spectrum, you would have longer regions like the polyglutamine from Drosophila mastermind. Indeed, Drosophila mastermind fits perfectly in this tail of the distribution. So if you think about it, we have the protein sequences. We don't have any protein structures with those features. It means that most likely protein sequence regions with those features will never be crystallized because they do not fold in the way we expect for them to be amenable to crystallization eventually. And this, of course, also points to the fact that you can build a predictor, because you can train a machine learning method, for instance, and you can try to recognize regions in the sequences which behave in a strange way. And in fact, this has been done by a number of people. The prediction challenge itself is quite simple. So you will have as input a protein sequence, and the output of the program would be a simple two-state labeling where you say for each residue, whether it's predicted as disordered or predicted as structured. And ideally, you would be able to pick up the regions which are disordered and which correspond, for instance, in this crystal structure here to these parts which are highly flexible. Now, of course, there are a few things to consider here. So how do you derive those data sets which are truly regions which are disordered? If we don't have an operational definition for disorder and we are just looking for something which is missing and which we cannot define, then this may be a challenge. However, what came out very early on is that you can take as proxy for disorder the absence of residues, so the so-called missing residues in crystal structures deposited in the protein data bank. And this has been employed very effectively. And in fact, one of the earliest methods to do this was Isopred, published by David Jones in 2003 and then in 2004. Over the years, there have been a number of other predictors doing machine learning along the same line. I will just briefly introduce how Isopred does it. So the protocol is very similar to Cypred, which was previously developed by the same author. And the analysis starts from a PsyBlast profile. So you take a sequence, you run a database search, and you see which are the homologous sequences, and you use a local window of 15 residues. And then you use the neural network, which was trained on recognizing disorder, which produces an output value between 0 and 1, where 0 means definitely structured, 1 means definitely disordered. And the network had been trained on 715 proteins of known structure. So this was actually one of the clever intuitions from David at that time, that he was taking the missing residues from the protein data bank as a proxy for disorder. And this has been working quite well. Of course, it's not the real thing. It's not the things you would pick up from the SwissProt sequences via the SEC program, but it comes pretty close, and it is a good definition. So then there have been many other predictors in this area. We've also had our go at this problem. The latest version of what we've been developing is called eSpritz and was developed by Ian Walsh, who's pictured here, who was a postdoc in our group at that time. And now he's a researcher in Singapore. So essentially, this was, again, a machine learning method, nothing special there, perhaps except the topology of the neural network which was used. The two key points here I wanted to make are, first of all, that it was trained on different flavors of disorder. So depending on how you train the method, you can be detecting things which are slightly different. So the missing residues from X-ray structures is one alternative. Another one would be Disprot, and I'll be talking in a few slides about the Disprot database. And then we also had an attempt to use NMR mobility data. The second thing, which is shown in this figure here, which is interesting, is the time required to compute 1% of the human genome. Now, one of the things taken for granted in the machine learning field for bioinformatics, when you try to predict any feature related to protein sequences, is that if you do a database search, you usually improve the performance. So everybody does this. However, if you run a database search, even a simple BLAST, against the likes of Uniprot, it takes a couple of minutes to be carried out, usually. And therefore, it makes for a much slower method. So what we looked at here is on the x-axis, the time required to compute 1% of the human genomes in terms of predictions. And notice that this is a logarithmic scale in seconds versus on the y-axis, the AUC, the area under the curve, which is one of the most popular performance measures. And sorry. And of course, as expected, the methods which leverage this evolutionary information through the multiple sequence alignment perform slightly better. However, what is really striking is that you can save 3 to 4 orders of magnitude on your computation time by having a method which works on a single sequence, and it performs almost as good as the very slow methods. So you're losing around 2% of AUC. And 2% of AUC is probably acceptable for many applications. Now consider, if you save 3 or 4 orders of magnitude, it means that you can much more easily do calculations on entire genomes. And in fact, this is something we will be coming back to, and this has led us to develop over the last 10 years almost the Mobidb database. But these are some words about the predictors. Let me now tell you a few things about the databases for IDPs, because predictors are nice, but they require data on which you can train them. And remember that we still have this schematic here where we don't truly know how to separate the various cases. And so it's, at the very least, just, yeah, we want to say that something is disordered as opposed to structured. So one database which has been around since 2005 is Disprot. Disprot was initially developed by Keith Dunker in the US in conjunction with Peter Tompe in Hungary at that time and some other co-workers. So the first publication was in 2005, and it had been hosted for about a decade in the US. And eventually there were funding issues, funding was coming and going, and so it had been lingering for a while until in 2017, we built a consortium around the Disprot database, and it was moved to our lab at the University of Padova. And we moved the annotation for Disprot into a community curation model. So we now have about, I think, 15 different groups in as many different European states who are contributing to the curation. And we have on the latest version, I think, something like 60 different curators contributing. So 2017 was a complete technological makeover of the database, and also the complete reannotation of the existing entries and 200 new entries. So it went to about 800 proteins Then in 2019, we put up DISPROT 8, which has a new format. It has a new ontology to describe the functions of disorder. And it was integrated with EuroPMC and some of the Elixir core data resources, and it doubled in size, essentially. Today, we have an even larger database of at least 2,000 entries. Some of them are embargoed for reasons I will be explaining in a few slides from now. So this is a manually curated database. And one of the key features of DISPROT is really that any statement about a part of the protein being disordered is backed up by a scientific publication, which you can find in PubMed. So this overview here shows you, again, P53, because it's one of the easiest examples. This C-terminal part here has been annotated as disordered in DISPROT, and the functional the functional region annotation is shown down here, where you have the ontology term, the region position, the detection method, and this is backed up by a reference. This is the PubMed identifier, and you can click on it and go into PubMed and actually read the paper and see for yourself the evidence supporting that this region is truly disordered, was identified like that. So it's a relatively small database, but it is the data is of high value because it has been curated. And the DISPROT evidence here, you see a better distinction of what is in the various entries. So we have the ontology terms, more about that later on, about the ontology. The fragments of which part of the amino acid sequence is involved. The method used to detect, the experimental method used to detect disorder, who the curator was for this, the statement from the paper saying that this is considered intrinsically disordered, the cross-reference where available, and the paper where the statement comes from, so the scientific publication. Now, DISPROT collects a number of these instances, and so at the end, it has several branches in the ontology. So there is a consensus mechanisms whereby regions of the same type, so the same ontology branch, are merged, and the consensus is generating, giving priority to interactions and structural transitions, then disorder, then order, and finally function, and this gives for a more complete view. Regarding the possible functions of disordered regions, this is a very simple schema which was originally proposed by Peter Tompa in 2005, where the 6 basic mechanisms of disordered regions would be, on one hand, entropic chains, which are loosely behaving like this cooked spaghettis in water. And then they could be elements of recognition for other molecules with either transient or permanent binding. And depending on whether the binding is transient or permanent, you have different subtypes. And this was the first attempt to try to categorize the function of disordered protein regions. This has been developed in DISPROT into a fully fledged disorder ontology, which is the IDPO. The current version, which is available, has 5 main namespaces or branches. And the detection methods are used as evidence codes. So 4 branches provide the annotation, and 1 is just the evidence code telling you which the detection method was. So for the detection methods, now we are moving to the Eco Ontology, which already contains a lot of the things we needed, and some additional terms are being developed in collaboration with the Eco team. The remaining 4 branches, that's the structural state, the structural transition, the interaction partner, and the disorder function. The most important one here is really the disorder function, because structural state is essentially whether it's ordered or disordered, or eventually if the disorder is a molten globule or other things. Structural transition is how it switches between its different states, and interaction partner is what is being recognized by the disordered region. So here you see some examples. So these are the various branches of the ontology, and this is a histogram of how many proteins and residues you have in the database at that time with a particular annotation. So you see that in terms of interaction partners, protein binding is the most common one. Then you have nucleic acid binding, iron binding, lipids, and small molecules. So it's fairly broad categories. Really, the disorder functions are the interesting part here, and more about that later on. And another interesting feature of this prot is that while it has a number of shorter disordered regions of 10 to 30 residues or thereabouts, these are quite common, it also contains a lot of long disordered regions above 100 residues. So these are 100 consecutive residues or more, which have been all determined experimentally as being intrinsically disordered. And these are very likely the functionally relevant disordered regions. So this is just an example for the more technically minded among the audience of how the protein is encoded as an entry object in this prot. So it has a unique this prot identifier and the accession number from UniProt, which is always mandatory. And then it has information about the regions and the consensus. And of course, it has the information about the annotation, which has been done, and the evidence codes, plus a number of other statements. And these can be queried also programmatically for those who want to do large-scale data mining on the database. So this is one approach to the database. Then we have another one, which is Mobidb. So you can think of the relationship between this prot and Mobidb pretty much like what UniProt does between Swissprot and Tremble. So the idea for Mobidb is to have a large-scale annotation of disorder according to this data quality pyramid shown here. So, of course, you have curated data, which would be the likes of this prot or a few other specialized databases. Those are of high quality, but they are relatively few. And so they get you only so far if you're interested in annotating all of UniProt, for instance. This would be the highest quality. Then below this, you have sort of indirect sources. So mining the protein data bank for regions with missing residues and crystal structures is an indirect way telling you that perhaps it's disorder, perhaps it was some other artifact. This is why it's called indirect, because it doesn't demonstrate disorder, but it suggests that it could be disordered. And that type of information you have in greater abundance, because the curated data, this is on the order of a few thousand proteins at most. The indirect sources, especially if you combine them also with homology transfer from this prot and the likes, you can have for tens of thousands of proteins. And of course, this is lower quality, but you have much more. And the lowest level down here would be the predictions. Of course, you can run predictions for all of UniProt, so over 200 million sequences. And of course, these are predictions, so they are not the real thing. They are not experimental evidence. They have to be taken with a grain of salt, but at least they provide annotation for all those proteins which have not been curated by an expert. So for all those over 200 million sequences where only Interpro is essentially contributing the annotation in UniProt. Mobidb was initially developed by Tommaso Di Domenico, which was a very clever PhD student at that time. And then it has been going on for almost a decade now. So the data provenance of Mobidb is actually from several sources. At the top layer, the curated information is taken from a handful of specialized resources. This prot is the largest of these contributing databases, but it's not the only one. The derived data is essentially a parsing and an analysis of the protein data bank. So this is leveraged from PDB Europe and run with some in-house algorithms to derive this kind of information. And at the bottom level, for all of the UniProt sequences, we can run the Mobidb Lite consensus predictor, and this provides the lowest tier in the quality pyramid. If you want to see a much more detailed picture, this is for the latest release of Mobidb, which was just published in Nucleic Acids Research, the database issue, a month ago. It has become quite sophisticated by now. So you see that we have a number of member databases where we take out information. The primary databases we're using, well, that's mostly the PDB at the moment, but interpreted with different algorithms. And then the protein sequences, well, this is run on UniProt KB with different types of predictors. So Mobidb has nowadays these 4 levels, because you have the curated data, the homologs to the curated data, the derived data, and the predictions. And Mobidb is also available through an API for programmatic access. And in fact, we have a few power users who tend to download tens of thousands of entries for their large-scale analysis projects. We had a couple of instances where someone actually downloaded millions of sequences in a single day, and we are very proud that the server worked very well, and we didn't even know when this was happening, except for the logs telling us that there was this peak in activity. Of course, Mobidb has then been also integrated with a number of other databases, most notably Interpro, UniProt KB, and PDB Europe. So there are different flavors of disorder which are being annotated. This is essentially plain vanilla intrinsic disorder, but also the linear interacting peptides and the more dynamic structures. Let me say a few words about those now. So the predictions, this is fairly standard. So we use a bunch of predictors shown here. And we report the prediction scores for each of them. This is a simple 2-colored scheme where this light blue means predicted as structured, orange means predicted as disordered. And this is then put together in Mobidb Lite. The curated is shown again with a similar feature viewer, and here you have an instance of a protein where you see different databases contributing different types of annotations. Of course, Gene3D is an annotation for structured domains and not disorder, but that is again useful. And this is combined in a consensus. And then, yeah, the derived annotations from the PDB, well, these are mostly missing residues in crystal structures, but they can be also regions with high temperature, so high B factors. And also in the case of NMR models, we can have mobile residues, which are detected with specialized algorithm. And these again are put together in this kind of consensus view. Dynamic structures, we have annotations for, again, this is also predictions for helix propensity, beta sheet propensity, and coils, and also where possible polyproline 2 helices and whether parts are considered rigid or flexible. Yeah, the predictions are calculated on all of the UniProt KB sequences, and for the features, it's provided by our fast secondary structure predictor, which is called FAS, and also Dynamine for rigidity. And here you see the current version of the Mobidb interface. Notice that this is the simple view for P53. And the key feature is really here in the middle. This is the feature viewer, and this is where all the information is summarized in a similar fashion to what you would find in the likes of Interpro. Sorry. So on the top part here, these are the interactors of P53. So you can click on them and you can quickly navigate and see how those interactors are for disorder. And then there's a section with cross references. And notice I was saying this is the simple view. So we try to make it as compact as possible, the feature viewer. The advanced view is this for P53. So you see there's a lot more of information. This is, of course, only relevant for those who really want to drill down and look at specific features of the protein sequence of interest and try to understand the finer points about whether it can be disordered or it can be annotated with different flavors of disorder in a database or indirectly through the PDB. And when there are PDB structures, you can even have this kind of view here where, especially this is useful for the likes of P53 and the short peptide bound to its interaction partner, because you can see which residues are really interacting and how they are interacting. So there's a lot of information, but this really requires that you would have to look at them to understand better the finer points here. Finally, although this was done on the previous release of Mobidb and of this brought, an interesting analysis we made is the overlap between the various databases curating for disordered regions. These are a few Venn diagrams. Notice that this is DISPROT, which is overall the largest database. And notice that the overlap between the various databases is very low. So it means one of two things. Either that the databases are really defining different things, which can be reasonable, but only up to a certain point, or much more likely what it means is that there are many more proteins out there which are intrinsically disordered and which have not been annotated in the database. So in fact, since then, for DISPROT, we have been targeting these proteins which were in other databases. So DISPROT is now larger and covers more of the things which are known from other databases to be disordered. A simple word about how you can transfer curated information by homology. So here you have sorry, a very simple case, which is P53 human. So P53 from human is present in DISPROT, and there are experiments done on this model organism demonstrating that a part of the protein sequence is disordered. But then you may be interested in P53 in mouse or rat. And chances are that neither P53 from mouse nor the one from rat or your other favorite mammal, say cat or dog or any other, they would probably not be in DISPROT because it's unlikely that someone did the experiment there. On the other hand, P53 in human, mouse, rat, and cat and dog are probably extremely conserved. So you can expect at most a few mutations. So any information you have for disorder on P53 human, you should be able to transfer to the mouse, rat, cat, and dog variant. And this we do by looking for the cases where there is very clear orthology. So we have only orthologs taken from gene tree, and which have the same protein architecture and have over 80% sequence identity. This is a sort of automatic inference, but it allows us to leverage the data from DISPROT. And this was still a previous release with about 800 proteins. We went from 800 proteins annotated in DISPROT to about 10,000 with this predicted DISPROT annotation via homology transfer. So it makes for a good difference. The other point is Mobidb Lite, which is the consensus predictor we are using. And this originated from a discussion I had almost 10 years ago by now with Alex Bateman, the head of PFAM and Interpro and in Uniprot at that time, who was saying that they were looking for something, for a fast predictor which would define the opposite of structured domains. So they wanted to have something able to detect long disorder, meaning at least 20 consecutive residues which are predicted as disordered. But they also wanted a very conservative predictor. So something which reports disorder only if it's virtually certain that it will be disordered, but otherwise it does not report it. And one of the problems with most of the disorder predictors is that they tend to over-predict quite a bit. So, sorry, the histogram you see down here is for various component predictors which are integrated into Mobidb Lite. In red, you have the average over-prediction of disorder, which is quite substantial, at least, especially for some of them. And in blue, you have the average under-prediction of disorder by those methods. So this is Mobidb Lite, which is just a simple majority consensus. So we combine the scores and we see whatever is most likely. And we only take the cases where you have at least 20 consecutive residues predicted by this large majority of methods. The result is a predictor which has almost no over-prediction, so almost no false positives. And this comes at the expense of having a higher fraction of false negatives. So we are missing out on some disorder, but whenever Mobidb Lite predicts something as disordered, it is a very strong statement. And this kind of approach has been taken up then by some of the core data resources. This was work done within Elixir. So since IDP annotations are significantly underrepresented in several Elixir core data resources, starting from Interpro and Uniprot, what we did as part of an implementation study there is that Mobidb Lite was integrated into Interpro. So nowadays, for the last couple of years, if you run Interpro, you also get results from Mobidb Lite, telling you which parts are disordered. And here you see a nice example for P53 and how the disorder predictions from Mobidb Lite are complementary to the regions identified by the other domain-based predictors, which are part of Interpro. And this type of information has then been also percolated into Uniprot KB and into the Protein Data Bank in Europe. This was again work done with several people at the EBI, Alex Bateman and Samir Valenkar, and for the SIB part of Uniprot, it was Alan Bridge coordinating this. And so now you have annotations, and this is the Prot Vista annotations for regions, which they call flexible again. So this has been integrated up to a certain point. Elixir, Rita also already mentioned this, it's the European infrastructure for biological data, and it has a community for intrinsically disordered proteins. And the key goals here are to develop the standards, the tools, and the resources to drive the integration of IDP data into the major databases, but also to drive the definition of additional types and to really make sense out of all of this. And one of the key elements in this is standardization. So in particular, Balint Mezaros and Norman Davey have been instrumental in driving the development of a common standard of minimum information about the disorder experiment, which will allow also the easier interchange of information between the participating databases. So that at the moment, what we are up to is that Several of the databases, notably Disprot, Mobidb, but also some other databases, will be all contributing to a central repository, which will be called IDP Central. And that this will be the portal for information on disordered regions to bring it into the main databases in the Elixir ecosystem. So finally, on this part about databases, I have just one slide saying that we've also been trying to tackle the structural ensembles behind intrinsically disordered regions. And this is the PED, the Protein Ensemble Database. This has been around since 2014, I believe, and it was hosted in Brussels by Peter Tompa's lab. Now it's a collaboration, so the technical part is again carried out in Padova by our group, and the rest is a joint venture. And it is a database where you can find ensembles of structures which are derived according to experimental constraints from NMR, SACS, or FRET data. But these are all simulated ensembles. So they obey the experimental constraints, but the experimental constraints are relatively weak. They give you a few hints about some distances between pairs of residues or things like that. And so one of the ongoing pieces of work here is to figure out how useful these ensembles can be and how realistic they are in representing what is the true behavior of these disordered regions. On the other hand, PED is a deposition database, so now we have one place where these ensembles are being collected, and this is an important step to try to drive the field forward. So that the only remaining part I wanted to mention about this is, sorry, Rita, how much time do I have in total? We are supposed to keep time for questions at the end.
For question, yes, but we have as much time as you want. We are supposed to finish at 5, comprising the
no, because I have
so.
3 separate modules now, so I can also either do them all or I can stop earlier, depending on how much time we want to keep for questions. So,
in principle, you have 40 minutes from now, so let's keep at least 20 minutes for questions.
Okay, cool. Okay, so I will be talking briefly about this part about the linear interacting peptides. So again, the concept about the different types of disorder you have here, which can have different features, this is quite intriguing, and we are only starting to grapple with this and to try to get a feeling of what is already out there and what we can also try to extrapolate from the protein data bank, because remember that there's a lot of data in there already. So a related view of this is shown in a recent paper by Monika Fuchsreiter's group, who tried to classify the different types of behavior you can have. So again, we are dealing with things which are either disordered or disordered and become structured, or things which can be always disordered, and yet they interact with something else. So these are the so-called binding modes of disordered proteins, and these sample a very wide spectrum of structure and dynamics. And here you see different examples. So on one hand, you have the short linear motifs, which you have a short fragment of the protein fitting into this binding groove here and undergoing a disorder to order transition. So once bound, this is structured. It's kept in place, and then when it is released, it becomes unstructured again. But you can also have cases where the binding is not as well defined and there is some variability, or the binding can be context dependent, so there is some changes on how it binds on the surface of its partner protein, or it can be things where it's a type of fuzzy binding. So these two come together, they like each other, so they do interact, but it's not well defined how they interact. And so one moves around on the surface of the other. And finally, you can have cases which are the most extreme example of dynamics. Here you have a disorder to disorder interaction. In this case, one of them is completely floating around and the other is also moving, and yet they stay in contact somehow. So these are different cases. And in principle, this type of data is also available from the PDB and can be leveraged. So two examples here is the disorder to order transition P27 has this alpha helix here, which is stable in solution. The rest is disordered. And then when it binds, it can form this structure here, wraps around. Disorder to disorder would be this distance here, when you have a lot of different conformations. And context-dependent binding is here, for instance, P15 PAF, which, depending on its binding partner, changes the way it is. So one idea here is to try to see how much of these different instances you can really leverage from an analysis of the protein data bank. So how many of these cases are already there and we just haven't had a proper look at them. And this is why we came up with a proposition to look for linear interacting peptides, or what we call LIPs. This is roughly similar to the concept of short linear motifs. So something binding on the surface of a globular protein domain. The key concept here is that linear interacting peptides have more interactions with their binding partner than with themselves. So it's not a well-folded globular protein which is binding to something else. It is something which really requires all the interactions with the binding partner in order to be stable. And this is something which can be recognized by a large-scale analysis of the protein data bank. So we're looking at fragments which are on the order of 20, 30 residues, perhaps. And some of them are manually curated in databases. The others, I mean, a thing like this one, you can recognize by just looking at the protein structure. But of course, you cannot ask someone to trawl through over 100,000 proteins, which are now in the PDB. You need an algorithm to do that. And our attempt to do this is called FLIPPER, which is a fast linear interacting peptides predictor, which extracts various structural features from the protein data bank in files. And it tries to automatically learn a function to classify residues as LIPs. It uses a random forest classifier with an ensemble of 20 decision trees. So we try to use a very simple machine learning method because these are usually more robust and we don't want to overfit on the data because there are relatively few instances which are manually created at this time. So we really want to have something which can help to look for more cases. The features which are used in FLIPPER in order to try to classify are the contacts, which is essentially if two residues are considered neighbors, if the two closest atoms are less than 3.8 angstrom of distance, and these can be interchain, so with itself, with a binding partner, or intrachain, which means with itself. We also look at secondary structure, relative surface area, linearity, which is a function of straightness, because obviously if it's straight and it has a greater ability to interact with something else. If it's completely folded on its own, then it's less likely to interact. And the chain length is also a parameter. This has been trained and benchmarked onto data sets I will not describe in more detail, which is PixelDB and DIPS. The Matthews correlation coefficient for FLIPPER is quite good. So in most instances, it's over 0.94. 94 for the DIBS dataset, which is somewhat different. It's 0.85. So it essentially is able to recognize something which is the clear cases annotated from the data sets where these interactions are. So then we also figured out that we should be trying to split this into different categories, and I will show you just a brief example for each one of them. One is homo interactions, so consider two protein chains of the same type. Then you have the hetero interactions, which can be either with another protein, which is different, or nucleic acids, or a mixture of protein and nucleic acids. And these are some numbers of the different types which are being recognized. I will skip the details. And just to give you examples of what we are able to pick up with Flipper. So these are the regions being picked up in this long tail fiber protein. So you see that this is really a dimer, and we recognize this as being something which interacts. This is the RNA binding protein FAS, which forms these aggregates, and so these are also picked up by Flipper. And finally, P53, this is one of its binding regions, and you see that this is also picked up, and this is interesting information. Now, of course, so far, I haven't really told you whether these are disordered regions or not, because it's not part of what we've been trying to do so far. So you should consider Flipper just as an idea generator for things which will then be curated for Disprot, for instance. Here you see other examples. So this is P27, which is picked up, and this is a nucleic acid binding protein, and this is a mixture of the two. So the usage we intend to have for Flipper is to have this as an idea generator for curating data in Disprot, but also to have this in Mobidb, where it's already available, where you can try to look for instances where these could be interactions with other binding partners, and you can decide for yourself whether you consider them to be intrinsically disordered or not. So it is a way for experimentalists, for instance, to try to gauge whether it's worthwhile to make an experiment to see whether that region is truly disordered or not. Finally, and I will just say a few words about this, one of the things we have been doing, since we have this large data set of curated data, which has relatively high quality, so Disprot is really the gold standard in the field for intrinsically disordered proteins. And once you have one of these nice data sets, you can also run critical assessments to try to see how well the predictors work. And this is KIDE and CAFA. So for predicting the disordered regions, there have been a number of attempts to benchmark the predictors. The CASP experiment, so the critical assessment techniques for protein structure prediction, has been doing it for a decade, between 2002 and 2012, but then had to stop because they did not have good targets, because they only had the missing residues from the protein data bank crystal structures, essentially, and those were not the really functional disorder. So on the other hand, what we could do is to do a blind assessment on all the available predictors at that time, 2 years ago, with a data set which was still unseen by any of those predictors, because these were the newly curated Disprot entries. So KIDE has been mostly done by these two people here, who are Marco Necci and Damiano Piovesan, a postdoc and a researcher in our group. And as ground truth, we use Disprot. The prediction software is installed locally on the Mobidb servers, and we run the assessment both for accuracy, which is the traditional way to do it, and also for a technical evaluation. And by the way, for those interested, this is the URL, so idpcentral.org/kite. The prediction setting is that this is essentially a regular Linux cluster, and we installed the software locally. We had a lot of unexpected features. So one of the things we did not envisage is that there are a number of software engineering issues with many of the predictors. And in fact, only one predictor was provided as a container or virtual machine. So there was quite a bit of work to do. Some of them were also quite complicated to install. But in the end, we had 43 outputs, so 32 disorder predictors and 12 predictors for disordered binding regions. So the likes of things which could be picked up with Flipper, for instance. So then regarding the classification of what is distorted and what is structured, I will not go into the details. In principle, we have two separate definitions. These differ on whether you consider only as structured what is available in the PDB or also what is not annotated as disordered in Disprot. There are arguments for both of these. In the end, we believe that considering what is not annotated as disordered in Disprot as structured is probably more valid. But this is there is an ample discussion of this in the paper we are publishing now. So the results are shown here for the Disprot data set, and it is encouraging in the sense that the predictors perform better than all the baselines we have, which is very good. And still there is room for improvement. This is the most difficult type of assessment we can do. And therefore, we believe that there will be a newer generation of methods which can still perform better. And these are being developed at the moment. Then one thing we could do is also benchmark for the first time the fully disordered proteins. And it turns out that there's a lot of variability in there among the predictors. Regarding the the disordered binding regions, the predictors perform much worse than before, and in fact they perform only slightly. The best perform only slightly better than the baselines. So here there is really a lot of room for improvement. But this is essentially due to the lack of a good training data set, which is precisely the reason why we came up with Flipper in order to try to drive the definition of these data sets. So eventually, there are a lot of interesting considerations. The paper itself is currently available as a preprint in bioRxiv. This is the URL if you want to look it up. And the paper has also just been accepted about a week ago in Nature Methods for publication. So it will be coming out, I believe, in the April issue of Nature Methods. But you can read the preprint, which really contains all the information. So finally, we are about to run a new round of KIDE. So we are currently in the process of collecting new predictive software. If, in case you have anything you want to contribute, please go look and see the idpcentral.org/KIDE webpage for instructions on how to contribute. Just very, very briefly, a few words, because we've also been contributing a challenge to CAFA, which is the Critical Assessment of Function Annotation, where we ask predictors to give us predictions for the IP ontology of this prot. And so this is the regular CAFA timeline. The preliminary results were published. Not published. They were presented at last year's ISMB conference. So at the moment, we are waiting for the organizers to give us the green light for the final assessment so that we can write up the paper. And this is, again, the Disprot annotation. Remember that we have these ontology terms. So these are a novel ontology of functions related to disorder. And there are several of these. It's still, of course, much smaller than the gene ontology molecular function component, but it is there and it has a number of entries. So there are different categories. This is an example of a term which is flexible linker. So these are essentially regions which modulate the distance between domains, but they don't bind to anything per se. We have a molecular recognition effector. So these are things where the binding is of that disordered regions is necessary in order to carry out a specific effect. Tethering is if it serves to keep something connected to another part of the complex, and there are several others. Okay, targeting, let me skip over those. This is just a data set composition between the ground truth and the training set, which was made available to the predictors. And you can see that the distribution of terms used in DISPRA to annotate is quite skewed. So flexible linker or spacer is the most common one. And for those who are familiar with kappa, this is our equivalent of the molecular function term binding, which is very common. So then there are a couple of others I will not address further, which are quite common. The assessment was carried out with the regular kappa assessment tool. And yeah, just very briefly, this is the typical kappa output plot for the groups which have been participating. Again, those familiar with kappa will notice that the performance here is not as good as in, say, the molecular function branch of gene ontology. So there is still quite a bit room for improvement, but they are doing better than the baselines, which are in these dotted lines here. So there are some methods which really are able to do some sort of prediction. There were not too many groups participating. And then we have carried out the same analysis without the term linker, which was the most abundant one. And you see that the performance drops. So clearly currently predicting things to be linkers according to the IDP ontology was giving some methods a boost. But still, there is signal, although there's quite a bit room for improvement. But then again, this is the first time that people were asked to build a predictor for this type of challenge. So we envisage that there will be more of these predictors coming out over the coming year or so. So finally, let me acknowledge all the people in the lab who have been contributing to this and the various consortia we're involved in. Mainly, I want to thank Dr. Damiano Piovesan, who is also shown here, who is the senior researcher in the group and who is really coordinating most of the projects I have been talking about today. And finally, if you're interested to hear more about intrinsic disorder and related topics, so if you really can't get enough of this topic, then we are organizing as part of the IDP Fund Consortium a series of webinars, which are once per month. You can register for them at this URL here, which is idpfund.eu. And the next webinar is due in slightly more than a week from now. And we have Peter Tompa talking about structural disorder, motives, and domains in linear and liquid-liquid phase separation, which is quite a hot topic. And also we have Tadeo Saldaño from the University of Quilmes in Argentina to talking about exploring conformational spaces of proteins by normal modes analysis. And with that, I would like to thank you for your attention, and I hope we have enough time for the questions.
Thank you, Silvio. Thank you for the lecture. Time is open for questions, so you can raise your hand. You can raise your hand from the reaction button. I invite all the students, of PhD students and the master students to ask questions. I can start with maybe I missed. Is there a relationship between the amount of disorder in a sequence and the number of interactors? I saw that in the last version of Mobidb, you put the interactors and the disorder prediction.
Yes, let me go back to some slide related to this. Yes, Mobidb, the interactors we have here are taken from, okay, these ones.
Yes.
These are taken from, I believe, UniProt, because we take all the information on the top part on the fly from the UniProt database. It's a very good question. There are some studies which have been published a number of years ago which do try to establish this relationship. In general terms, yes, but the studies were done on predictions, and so again, they have to be taken with a grain of salt. But one of the things which are thought in the community is that disordered proteins should on average have more interactors, and that most of these interactions are transient. And for instance, a lot of proteins involved in cancer are enriched in disordered regions. So this is also, there is a logical reason for this, because proteins in cancer tend to be the ones who regulate the behavior of the cell. And as we've seen, regulation is a type of function which is correlated with disorder. But again, these are, for the most part, correlation studies. So it would be nice to do this kind of analysis with more high quality data. So once we have a good number of proteins curated in this prot, we would like to carry out this type of analysis, because that would be, I believe, more meaningful than just running predictors. But yeah, in general, there is this positive correlation. Another area where intrinsic disorder is enriched are proteins of the central nervous system. Again, because these are very special structures which have to deal a lot with transient interactions.
Okay, see Rita. The microphone is off, sorry.
Well, I warmly invite students to ask questions, but in any case, I have a question for Silvio, and the question is just a curiosity. You mentioned the eco evidence in your database, yep. And then I was just grasping in your slides terms such as lack of electron density or NMR. Do you have other evidences or what? Because, basically you are referring to, well, NMR, it's a long story that in principle, whatever is water solvent exposed is somehow flexible by definition, or we have many cases NMR structures indeed are sort of, sets of different possible conformation of the protein, and basically they differ in the water exposed surface area. And then lack of electron density, well, depends very much on the method, on the resolution of the protein, etc., etc. So I was wondering if you have other eco evidence that you take into consideration in your database.
Yes, that's a very good question. So indeed, we have a few dozen different experimental techniques which are used in this prot. These are available on the DISPROT website if you want to read the different types we have. ECO itself, which is the evidence and conclusion ontology, is being developed, and ECO contains some of the basic techniques like X-ray crystallography and NMR. But what we are doing now with the eco developers is that they ask us to provide the initial definition of several of the techniques which are still missing from the eco ontology. So here in this plot, you see down here for the detection method, there are a few classes of things like spectrometry, optical analysis, analytical chemistry. There's actually more terms we have in the DISPROT database, and several of those are not available. So it's by far not just missing electron density. The detection methods. Let me just open this here. I have it. Okay, let me open all of them and let me move the share screen.
Doesn't matter. I will do it by myself. No problem.
Just to show you, these are all the terms which we have here, and there's quite a few of those. So yeah, and many of these are missing from the eco ontology. I think they will have quite a bit of work for a while. Okay.
Yes, thank you.
We have a question from Elisa Bertolini.
Yes, thanks. So it's me. So first of all, congratulations for the presentation. I think it was great. And also the models that are behind. So mainly one thing that I haven't understood really, the low complexity regions, and then you were talking about comparing it with the PDB structure, so to derive something that is not behaving as expected or predicted by the PDB model. So I haven't understood well this concept, and the fact that if there is a threshold on the entropy, which threshold are you taking, and if that can induce the results towards one. So it's an assumption that probably can cause certain results instead of others. So please, thanks.
Okay, that's a good question. Let me get back to the slide here, which you see with Drosophila mastermind. So low complexity regions are essentially things like this, polyQ and similar things. This is what you would like to pick up. So the very clear cases here, but then there are also other regions where it's a matter of defining a threshold. So would you consider this part, I don't know, here, where you have multiple glycines, but you also have leucine, asparagine, arginine. Would you consider this still a low complexity region? Probably. But then you have other sites where here, for instance, you have a lot of different amino acids, and this is a high complexity, and this is above threshold. So what SEG does is really just something which is very simple from the mathematical point of view. The formula here is just the Shannon entropy. This is very easy to calculate because it's just counting the positions and using a logarithm and summing over them. So it has a clear basis in information, in terms of information content. It does not have a clear definition at all in terms of biological meaning. So one of the things we have been doing is trying to sort out the complexities of low complexity definitions. We have a paper in briefings in bioinformatics from about a year ago, which was again developed by a group of people, where we try to collect different types of definitions and sort out between what can be calculated easily and what is the biologically meaningful definition. So there is not one clear threshold. there are, for instance, UniProt uses a relatively conservative SEG threshold to identify something as being, I don't know, glutamine rich. But there's an entire gray area of things where you cannot really say what it means that they are low complexity, or whether you should classify them as low complexity, or whether they are even disordered, because some of the low complexity reasons are not disordered. If you think about polyleucine, it may seem strange, but there are polyleucine regions in the various proteomes, and these are likely not disordered. They are hydrophobic, so they will be carrying out something, but we don't know exactly what the structure would look like because it's not available in the protein data bank. So there are some of these cases, and again, I can point you to the review there as a place where we discuss all of this. So the advantage of SEG is that it's very easy to calculate. It's very fast, but the threshold you use is highly arbitrary. I guess that is my key message.
Thank you very much.
Okay.
More questions? Students?
Then I have another question in case.
You're allowed, of course.
But
Since all the students seem to
somebody
be...
from the audience is coming up,
Yeah.
it's okay, because I don't want to take advantage of the situation.
Go
Okay,
ahead, please.
in the meantime, I will ask my question, and the question is, at the beginning, even with P53, you were showing us situations in which evidently the shortage of the segment of the protein that is in contact with some other protein or is co-crystallized or I don't know, bound to another protein is short. And my question would be, when you have short segments, so fragments around, even when you are producing or you are co-crystallizing short segments together with a specific protein, it may be possible that the that the conformation that is somehow that has been frozen during the crystallization process is due to a pure interaction that is somehow due to the solvent exposure to the protein ligand interaction, etc. So, because when you have the whole length protein, that's fine. I may agree with you that you may have different architectures bound to the same protein, but in many cases, in many instances, proteins are co-crystallized perhaps with short fragments. In that case, to know the whole structure of the fragment is a little bit awkward since you have only small fragments. And these small fragments indeed can adopt different conformations in solution. So your observation.
Now, my observation is at least twofold. So on one hand, this is why we curate in plot, for instance, because you cannot take things at face value from the protein data bank and expect that they will always be correct. So the key difference between this plot and Mobidb is that Mobidb provides this large-scale analysis, and you can find the things and you decide for yourself whether you trust them or not. This plot is a different pair of shoes because this plot we have curators which curate up to a quality standard, and they do read the papers, and if they curate something as being disordered, for instance, or one of these disordered to order transitions, then it means that there was evidence in the paper. So the experimentalists were saying or considering that this was disordered and yet binding to another partner. Nowadays, what happens with many crystal structures is that if you find, I don't know, protein X with a short piece of p53 bound to it in the crystal, then usually what it means is that the crystallographer was trying to elucidate the binding mode of that p53 fragment with that binding partner. Mm-hmm. But of course, you cannot take that at face value. There can be all sorts of different things. On the other hand, the protein data bank nowadays is already well mapped to UniProt. So if you have a short peptide, say 5 residues, usually the crystallographers, when they deposit this, already are saying which protein it came from. Mm-hmm. And in what we found in practical terms is that very often the fragment which is bound and is crystallized is part of a longer segment which they expressed but didn't crystallize the rest. So these are missing residues. And those are all the hallmarks of a binding region inside the disordered region. But of course, they need to be curated in order to be considered bona fide instances. Otherwise, it is a prediction and it may still be wrong.
Okay, thank you very much.
So, more questions? Elisa Bertolini?
Yes, if I may, sorry.
Yes,
yes, another question. Just a curiosity. So, of course, we are still studying, so we need to have experience with the models that are used and developed. But by curiosity, are you using as well some GLS analysis, so the generalized least square analysis, to confirm or infer relations of dependency or independencies among the variables that you've used into the models, or it's more to do with machine learning, neural networks, or other kind of models?
Okay, so we have not been developing any new machine learning method for the last couple of years at least, because what we try to do is just the consensus, which is Mobidb Lite, and improving that, but that fills a very specific niche. Regarding the machine learning, the machine learning methods themselves, these are usually benchmarked quite heavily, and the ones which perform well usually go to great pains to make sure that they are not overfitting and that they are really trying to capture the biological signal. But in our experience, one of the areas where there is still a lot of room for improvement is the data sets which are at the basis of the machine learning methods which are being developed for intrinsically disordered proteins. So just for comparison, secondary structure prediction is one of the oldest fields probably where machine learning has been applied to predictions from protein sequence. And even there, the neural networks were one of the key steps, of course. This is what happened in the 1990s. But one major contribution was the increasing size of the data sets on which it could be trained. So going from a couple of hundred examples to several thousands has inherently improved the performance of the methods. And in fact, since the year 2000 or thereabouts, the increase in performance for secondary structure predictors has more or less plateaued, or at least people have not been able to measure significant improvements, which may be also because it has become less fashionable, but and maybe now with the deep learning techniques there can be again a certain improvement, but that stage we have not reached yet for intrinsic disorder. So here, looking at the biology of the problem can still give you better data sets on which to train your methods. I guess that's probably one other take-home message from my talk.
Okay, thank you. If there are no questions, I thank Silvio again for the lecture, for being with us also this year in remote. We hope to have you in Bologna very soon. And I invite everybody for the next lecture on Monday from 3 to 5 in the afternoon. Professor David Jones will talk about artificial intelligence and protein structure prediction. So thank you again and see you soon.
Thank you.
Thank you, Silvio.
Ciao.
Thank you.
Ciao,
Bye-bye.
Silvio. Bye-bye.
Thank you. Bye-bye.
Ciao Silvio.